Now I'll introduce our next talk. We have Dr. Rangarajan, Chief Scientific Officer at Metabolon, along with a strong scientific expertise in translational sciences. He also boasts significant expertise in the deployment of AI analytics, machine learning integration to domain-specific RW environments for translation of early discovery R&D and pharmaceutical development. Today, he will dive deep into standardization for data connectivity, analytics, and actionable solutions with metabolomic data as a case study for target identification and validation of its potential clinical utility as a pathway for drug discovery.
Thanks, everyone. Can everybody hear me?
Yes.
Thank you. Thanks for the kind introduction, and I'm trying to share my screen here. So good afternoon, good evening, everyone. Thank you for this opportunity to speak. And I'm Ranga Sarangarajan. I am the Chief Scientific Officer at Metabolon. And, I'm not a computer programmer by any shape, size, or form. I fell into the data construct purely because of the need for analytics and the utility in understanding biology for the large volume data sets that we used to generate in my previous position. And when I moved into Metabolon, it provided a unique opportunity to look at data from a service side of things. And at Metabolon, we are a service company. Our name to fame is we are service providers for comprehensive profiling of human biospecimens and any biological specimens for characterization and profiling of small molecules. And then we generate the data, and once the data is generated, we provide interpretative services as well. And so today, what I'm going to be talking about is the kind of data that we generate and our small step in terms of building a structure that is consistent with what the FAIR principles are. And my slide is not moving. Okay, hopefully the slide moved for everybody.
Yes.
So I just wanted to level set. There is an explosion of data when we are talking about the concept of health and monitoring of health. And over the past few years, people have moved away from not only looking at just the molecular matrix, which we are fundamentally talking about the gene, the RNA, the protein, the fat molecules, as well as the small molecules, which is one of the constructs we generate. But how do you overlay that data sets across some of these other data modalities that are being captured as part of our understanding of health? And there are many permutations and combinations that are being looked at, but ultimately the ideal solution is which of these data streams that are outside the scope of what the molecular matrices are will marry together really well. And so one of the things that we have done internally is to start, over the past, this started long before I came in, but how do you generate foundational structures for the data so that we are giving or providing our customers the best foot forward in terms of data utilization? And so our construct primarily is, if you follow through the FAIR data principle, like I said, we are doing a very small step towards this FAIR principle. Is our fuel or currency that we have is the high quality data that we generate. And upon that, we also have a sizable metadata that we acquire from our potential clients so that we are able to connect those dots between the metadata and the metabolomic data that is being generated. And we had a fairly rudimentary portal until a few years back in terms of providing the data access to our clients. But over the last 2 years, we have also embarked on a very ambitious program where we are a massive update to our portal, where we will be able to provide them with all sorts of computational bandwidth for them to play around with the data on our own, with tools that we are building, so that they are able to use, leverage and use this data more meaningful. So just to level set for people who might not know what metabolomics is. So metabolomics basically refers to all small molecules. So the small molecules, the simplest example I can give is when we are talking about glucose measurement using one of those glucometers. Glucose is one of the molecules which would be considered a metabolite. Or also looking at the lipid side of things, if you go for a blood test, they are making measurements of the total cholesterol. So we profile the various shades of cholesterol molecules or lipid molecules and are able to provide that comprehensive spectrum. And so the small molecules, when you look at it collectively, there are multiple sources of it. There is the human one would refer to as a human exposome, where we are talking about the exposures from food, drink, air, as well as the small molecules that are generated internally from our just day-to-day activities of our systems biology that goes on. Plus, there are other things that we don't really pay attention to, at least on the day-to-day basis, the average person doesn't, but the gut or the gastrointestinal tract, starting from our mouth all the way up to the end of it, where microbiome or bugs basically inhabit the GI tract, and therefore there is a large consortium of bugs that produce small molecules themselves, but they also utilize and reprocess some of the human generated small molecules as well as those that are ingested. And so all those compendia of molecules are captured by a combination of technologies at Metabolon, and we generate this comprehensive profile. And these are massive data sets for each of these studies. And these molecules fit within the constructs of pathways. And basically what we do is, depending upon the client request, that the services provided, it may include also processing of these data and then providing biological insights and interpretation of those. So just to give you a scale of the kind of data classes or biological metabolite classes, on the left-hand side of the screen, what you are looking at are the various chemical entities or chemical classes one would encompass within the small molecular metabolome. But the previous presenter actually talked about a very interesting concept, right? The quality of the data and what does it mean. So the quality of the data and how good that is, it's also contextual, because in the world of metabolomics, the gold standards of a good readout of a small molecule is the ability to associate it with the corresponding standard that has been generated and characterized in the same manner of the data that is being generated. And at Metabolon, we have approximately 5,400 plus metabolites for which we have reference library, which is one of the biggest out there. And so every data output that we provide our clients, we are able to actually correlate all each of the data small molecule and their versions associated with a reference library standard that we then assure them basically that what we provide our clients is exactly what it is in their sample sets. And so it's a fairly comprehensive offering. But one of the other aspects that was referred to in the previous discussion is, how do you connect the dots? And one of the major components of our services that we provide is it possible to connect data that was generated on one time, and then if from on one experiment that was done now and another experiment done maybe a year from now, 2 years from now, 3 years from now, is it possible to connect the data cohorts? And the answer is yes, because when we run our samples for our clients, one of the biggest value propositions is we include certain reference standards within each of the data sets that we run that enables the clients to actually connect the data across cohorts. And so if somebody has done a study 5 years ago and they have used us for providing that service, and if they want to match or compare their results from a recent study, it is actually possible to do. And so that's one of the things that we have incorporated within our workflow. And then on the other side of the equation, so what I just talked about was on the client side in terms of the ability to connect the data from a analysis perspective on the study design and how the data across time can be connected. But the other side of that equation is each metabolite has so much of information and metadata associated with it. And so we are in the process, we already have a sizable information base that exists within the company that has been built over the years, but we are constantly updating it. And one of the reasons why we are now, and this is a plug with for Elucidata as well, in the sense that we have been actively discussing how do we work with third parties who do the data standardization much better than we do, so that we are able to focus on our strengths internally and outsource some of these activities. But the end goal or the ideal situation is to get into a common data ontological platform for all our metabolites, so that when we open up the portal for the client and they are able to overlay that data across this information that resides within our environment, and for them to make sense of how their data set is relevant against what is the information that resides out externally. And so it's all about enabling our clients to think about their data and in a hypothesis generating mode for them to think about the next steps, so that they are able to make useful interpretations of the data that is being generated. These were some of the very rudimentary stuff that is usually done in metabolomics, and the only reason I'm bringing this up is because I wasn't sure how much of the panel actually understands metabolomics or how the data is used. So some of the fundamental things for almost all the molecular matrices that one generates, whether it is the metabolomics, proteomics, or lipidomics, is the use of things like heat maps, pathway analysis, pathway enrichments. There are publicly available databases against which these are constructed and enriched for. And so these tools are readily available. But what we are trying to achieve here at Metabolon is we are building these massive human disease cohorts for that contains anywhere between a few hundred to a few thousand of varying disease etiologies, because the ultimate goal is, this is the overarching global goal, which is a multi-year strategic vision, is if we are able to collect samples, are able to bridge across these various studies, and has a common data foundation that is sitting on a knowledge base, it becomes very easy then to start looking at, say, if we have a cohort of patients with metadata associated with it, where we are able to get the multi-omic information on early manifestation of disease as informed by any sort of clinical benchmarks that are current. And then on the other end of that spectrum, we have patient cohorts with a very profound manifestation of the disease associated with the clinical data. Then metabolomics becomes a pivot point for generating, for identification of a panel of potential biomarkers that can then be cross-validated within the internal data sets that are generated. And basically, linking the biology to disease and the ability to start development of solutions that are actionable and can be translated towards the clinical, to a clinical or commercial value. And that's the overarching goal. And these are some of our recent initiatives where, just to give you a very brief idea on these initiatives of what Metabolon has embarked on. And just to give you this slide is more about the thought process rather than the actual numbers here. Human cohorts can be as small as about a few hundred, whereas several of the population health cohorts, which are present in foundations and academic settings, can reach several thousand or tens of thousands, and embedded within them, especially for chronic diseases like heart diseases, Parkinson's, Alzheimer's, or cardiovascular obesity. They are all polygenic, multifactorial, for which there are no real understanding of the correlation or the association between the genetic susceptibility to the actual causation of the disease. And so metabolomics, if you from a molecular matrix, is a unique in its ability to be a pivot point for all the biological functional endpoint. So whatever the genes, think of genes as a makeup of a template, and all the functionality, at the end of a biological run, metabolomics is probably a optimal readout. Yes, there are noises associated with it, but then again, once you reach a certain critical point in terms of the data size, then it is possible to actually come up with really tight endpoints. I should say, really, it's possible to run the analysis to identify panels of these small molecules that are pretty good in terms of prediction for certain clinical endpoints. And so, so that's based on this premise. We are investing heavily in making sure that the data that is generated, the data that is collected, the data that is curated appropriately and laid out in the common foundation, so that we are actually able to use this over a long period of time. So I think this is my last slide. So ultimately, in our small way, what we are trying to do is embark on a very ambitious plan where we are using a technology that is very well honed at Metabolon. We have 20 years of experience in developing the technology for generating some exquisitely reproducible data on metabolomics, and then also having the knowledge base that has been accumulated over the years on the standard platform, enabling our clients to actually leverage that information to be able to use the data sets that is generated as part of the services to then create the value out of it. I'm going to stop there, and I'm open to any questions.
Thank you, Ranga. That was really incredibly informative session. I think we have time only for one question, so I will start with the very first one that comes up. In principle, how does metabolite annotation work in Metabolon tool, and how accurate are those annotations?
I'm sorry, can you repeat that, please?
It's a long one, actually. In principle, how does metabolite annotation work in the Metabolon tool, and how accurate are those annotations?
Well, the annotations are as accurate as the information that comes out. So we spend a lot of time and effort making sure that our metabolites are annotated based upon our internal standards that we have, and all the associated information that is generated from our data sets, as well as everything that is available in the public domain. And so these are all cross-verified. And so we are fairly confident about the annotations of what is found, and then its associated linkage to biology, which would be in the humans.
Great. Thank you very much. Thanks for the talk.
Thank you.